Let's learn about eukaryotic transcription. Why we need transcription? The answer is here, as the proteins are the main building blocks of our body. Many important components in our body are made up of proteins, for example, enzymes and hormones. They are protein in nature, and similarly, many other more building components of our body are made up of proteins. But for the synthesis of these proteins, the special code and sequences are required. And this information and these instructions are stored in our body in the DNA. The DNA in our cells contains the instructions for building of all the proteins that make up our body. And these instructions in the DNA are present in the form of genes. How we can encode these genes? In order for a gene in DNA to be converted into protein, the process is required due to which the code has to be carried from the DNA for the protein synthesis. For the synthesis of RNA, this process is known as transcription. In transcription, the code is carried from the DNA in the form of messenger RNA, and then this messenger RNA is then used for this code is then taken to the ribosomes for the synthesis of the proteins. The formation of messenger RNA is known as transcription. Eukaryotes, the DNA is present inside the membrane-bounded organelle known as nucleus. So where the transcription takes place in the eukaryotic cells, as the DNA which has to be transcribed, or the instructions that are present inside the DNA which is to be copied in the form of RNA are present inside the nucleus. That's why the transcription will take place in the nucleus in eukaryotic cells. To understand the process of transcription, it is divided into different steps. The first step is the initiation. In initiation, as you can see here, the promoter region. In this step, the RNA polymerase enzyme binds to the promoter region and unwinds the double helical structure of DNA. Then it is followed by elongation, as the RNA polymerase keep on unwinding the double helical DNA and keep on adding the new nucleotide bases at the 3' hydroxyl end of the adjacent nucleotides. Then it will move forward until the terminator region reach. In the terminator region, there are specific sequences where the termination occurs, the transcription will stop and will release the RNA. In eukaryote, there is a specific process which is unique for the eukaryotes, that is known as RNA processing, or also known as post-transcriptional modification. In this process, when the RNA is released, it is premature RNA and need to be processed. After processing, it will become the mature RNA. But before going into detail into these processes, there is an additional step that is unique for the eukaryotic transcription, which is known as the formation of Preinitiation Complex. Before initiation, there is the formation of Preinitiation Complex, then the transcription will start. But before going into detail of these stages, let's learn about some more additional things that are required for the transcription process. What are these additional molecules that you must know about transcription? So the process of transcription in eukaryotes requires the three main things. This is RNA polymerase, transcription factors, and promoter sequences. Let's see one by one what are these. RNA polymerase. As RNA polymerase is an enzyme that catalyzes the synthesis of messenger RNA. RNA polymerase is basically the enzyme that will be responsible for the formation of the RNA chain. But in eukaryotes, there are three different types of RNA polymerases for the synthesis of different types of RNAs. Let's see here. RNA polymerase in eukaryotes are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1, it will catalyze the synthesis of ribosomal RNA. RNA polymerase 2, it will catalyze the synthesis of messenger RNA and snRNA. And RNA polymerase 3, it will catalyze the synthesis of transfer RNA and 5 subunit of ribosomal RNA. The other protein is these transcription factors. What are transcription factors? Transcription factors are the proteins, but they are a combination of different proteins. It is a highly complex structure. There are many proteins combined together to form the initiation complex. Let's see here which transcription factors are involved. These transcription factors are responsible for the recognition of the promoter region, as the promoter region consists of the specific sequences which are used for the recognition for the initiation of transcription. These transcription factors, together with the enzyme RNA polymerases, will bind to the promoter region. The promoter region, more specifically the Tata box and the Tata region, they will bind to them, recognize them. So that's why they are known as Tata box binding protein associated factors. These factors include transcription factor 2, D, A, B, E, F, and H. So all these factors together form a complex. In the transcription factor 2D, it consists of a binding region which is known as Tata binding protein. So this transcription factor 2D, the first transcription factor that comes and binds is the D factor, and it contains the specific binding region which is the Tata binding protein that will bind to the promoter region, the promoter sequence that is known as Tata box region. Promoter sequences are the sequences that are recognized by RNA polymerases. Where these promoter sequences are present, these promoter sequences are present upstream of the start or initiation point of the transcription. As you can see here in this diagram, this is the initiation point at +1, and then upstream to it, these are the promoter sequences bases. What are these promoter sequences? These are cat box, which is present 80 base pair upstream, GC box, which is present 100 base pair upstream, Tata box, which is present 20 base pair upstream, and then there is the +1 start point, or where the transcription initiation starts. So now you have learned about the RNA polymerases, which are in eukaryotes. There are 3 different types of RNA polymerases: RNA polymerase 1, 2, and 3 for the synthesis of different types of RNA. But most commonly we talk about messenger RNA, so that's why we have used RNA polymerase 2 in the process of transcription. Then you learned about different transcription factors. These transcription factors combine together to form the preinitiation complex together with the RNA polymerases, and then binds to the promoter sequences. And you have seen the different promoter sequences, that these are the specific nucleotide sequences that are present upstream of the initiation or start point of the transcription. Now let's start with the process of the transcription. Let's see one by one each in detail. Let's start with the formation of the preinitiation complex. In the preinitiation complex, the transcription factors along with the RNA polymerases combine together to form an assembly and form a complex structure known as the preinitiation complex. And this preinitiation complex recognizes the promoter region that contains the specific promoter sequences, and they bind to them. After binding to them, they provide the bending of the DNA molecule, and then after that the initiation starts. What are these transcription factors that combine together? These transcription factors, as I already explained, these different transcription factors combine together, but they have a specific sequence. When they come, they are present in the body as they are proteins, they are synthesized in the body. When they come to the promoter sequence, they come in the sequence one by one. So what is this sequence and how you can remember them? The sequence for the binding is transcription factor 2D, transcription factor 2A, as we are talking about messenger RNA, So the transcription factor and the RNA polymerases in this case will be the 2. And then transcription factor F, E, and H. So this is the sequence for binding of these transcription factors. How you can remember the sequence? There is a mnemonic for it. The mnemonic is dog, bark at people, frequently, every hour. By remembering this mnemonic, you can easily remember the sequence of these transcription factors. The dog, the D in the dog, D is for the transcription factor 2D, bark at, transcription factor A and B, people, P for RNA polymerases, F infrequently is for transcription factor 2F, every, in E in every is for the transcription factor 2E, and H in R is for the transcription factor H. So dog, bark at people frequently every hour. By memorizing this mnemonic, you can remember the sequence of these transcription factor bindings. Let's see how these transcription factors bind. The first factor that comes and binds to the promoter sequence, in 25% of cases, it binds to the Tata region or the Tata box. The first transcription factors that recognizes the promoter sequence is the transcription factor 2D. Because the transcription factor 2D, it contains the specific docking site for binding of this Tata region that is known as Tata binding protein. So that's why its name is TBP, Tata binding protein. This Tata binding protein, together with the transcription factor 2D, it binds to the Tata region, the Tata sequences, and then after binding, as you can see here, it creates a bend in the DNA structure. The other transcription factor that comes and binds transcription factor 2A and transcription factor 2B. After that, the RNA polymerase 2 is carried to the Tata region. It is carried to the Tata region by the transcription factor 2F. So transcription factor 2F together with the RNA polymerase 2 will come and bind to this region. Then it is followed by the recruitment of transcription factor 2E. Transcription factor 2E will come and binds to this complex. Transcription factor 2E, it basically provides the docking site for the transcription factor 2H. After that, as all these transcription factors bind transcription factor 2D, transcription factor 2A, B, F, E, and H together with the RNA polymerases, now there is the formation of the preinitiation complex. Now after the formation of the preinitiation complex, it will unwind the DNA. For the unwinding of the DNA, it requires the presence of the ATP. After the energy provision, after the utilization of the ATP, it opens up the DNA helix. When the DNA double helix opens up, here it starts the process of initiation. Now let's proceed with the second step, that is the initiation. After the formation of the preinitiation complex that is composed of the RNA polymerases together with associated proteins, these proteins are transcription factors. They bind to the promoter region of the DNA. They recognizes the promoter sequence and binds to them. Then after binding of this complex, it creates the bend in the DNA. After creating the bend in the DNA, there are activator proteins that will bind to the enhancer. So these activators act like a modulatory proteins which are involved in the regulation of the transcription by turning it on or by turning it off. In case of transcription, the activators bind, and in case of turning the transcription off, the repressor binds. RNA polymerase together with the transcription factors and associated proteins are responsible for unwinding of the double helical DNA. But at the initiation, first of all, there is the I formation and the breaking down of these hydrogen bases between the nucleotides This opening at first requires the presence of ATP, as it is energy dependent process. The initiation process is an energy dependent process. After that, the RNA polymerase will keep on moving forward and keep on adding the new nucleotide bases for the synthesis of the RNA. The RNA polymerases, as the strands move forward and it unwinds the double helical DNA strand, so as it moves forward, it breaks the hydrogen bond in the nucleotide. Similarly, at the same time, it keeps rewinding the previous nucleotide bases. They again, the hydrogen bonds form between them and again rewind them and to maintain the shape of the double helical DNA. For this purpose, the RNA polymerases at one time it only unwinds about 25 DNA base pairs. Only 25 base pairs are unwound at a time, and only 8 nucleotide base pairs will be attached by hydrogen bonding to these nucleotide bases. And the other RNA strand will fall off and the previous one will keep on rewinding and the forward will keep on unwinding. There will be forward will be keep on unwinding while the previous one will be keep on rewinding and again forming the double helical structure of DNA. After the initiation, let's proceed with the elongation process. What happens in the elongation? When the elongation process proceeds, as the RNA polymerases, when it unwinds the double helical structure, it keep on adding the nucleotide bases at the 3' hydroxyl. At that time, as you can see here, when it is moving forward, the RNA polymerases, all the transcription factors fall off. All the transcription factors leaves except of the one transcription factor, that is the transcription factor 2D, which contains the docking region tata binding protein. So this TBP, which is present inside the transcription factor 2D, it remain binds to the tata region until the whole process of transcription completes. When the whole elongation process and the complete RNA is synthesized, until then this transcription factor 2D along with the TBP will keep on binding to the tata region. How the process of elongation occurs? As the RNA is synthesized from 5' to 3'. As there is the synthesis of the strand, when the two double helical strands open up, the RNA polymerase breaks the hydrogen bonds between them, and when they are separated, the nucleotide bases are attached by the complementary base pairing. As complementary base pairing, the adenine binds with the thymine, but in this case, as this is the formation of the RNA newly synthesized strand, this is the copy of this DNA sequences, adenine will always bind with the uracil. As you can see here in RNA, there will be no thymine and the thymine is replaced with the uracil. And the rest guanine will bind with the cytosine. And then they form the hydrogen bonds. Adenine form the 2 hydrogen bonds while the guanine form the 3 hydrogen bonds. They will keep on binding and adding the nucleotides at the 3' hydroxyl end of the adjacent nucleotide. As the RNA polymerases, at the one time, they will keep on unwinding the DNA double helical structure and then keep on rewinding the double helical structure. To the downstream, they are moving, they are adding up the nucleotides until the sequences will reach that the RNA polymerases does not code for these sequences. These sequences are known as terminator sequences. When the terminator sequences reach, the RNA polymerase will stop and dissociate from this region. Now let's see with the final stage, that is the termination. What is termination? In eukaryotes, the termination occurs at the specific sequences known as terminator sequences. While the RNA polymerases, while copying the RNA, proceeding the transcription process, the sequences will come that the RNA polymerase will not be able to further catalyze the process of transcription. The RNA polymerase will dissociate This is the most common method, the first method, the sequences of the terminator. Most of the termination occurs by the terminator sequences. There are also other methods, that is the nucleosomes. Presence of nucleosomes may also lead to the dissociation of the RNA polymerases, and the termination will occur and will release the RNA free. And there is also the presence of some transcription factors that also helps in the process of termination, that will leave the RNA polymerase free and the RNA will be released. The final step, as the RNA which is synthesized is a premature RNA. For maturation of RNA, it has to pass through RNA processing. RNA processing, also known as post-transcriptional processing, or also known as post-transcriptional modifications. Let's see here. As the RNA is synthesized in the nucleus, it has to be released into the cytoplasm for the process of translation. To protect its ends by the degradation by exonucleases, the two processes are performed. One is the capping, this is the capping, and other is the tail addition. In the capping, there is the addition of the 5' 7-methylguanosine. This is the structure which is added to the 5', as the RNA which is synthesized is always 5' to 3'. At the 5', this structure is attached, which consists of sugar. This sugar attached by the phosphate bond and the guanosine base. And to the guanosine at the 7th position, the methyl group is attached. So that's why it is known as 7, because at 7 methyl is attached, and guanosine, this is the guanosine structure, and 5' of the messenger RNA, it will be attached. So that's why 7-methyl guanosine will be attached by the enzyme. 7-methyl transferase enzyme will be used for the addition of capping to the 5' and to the 3' poly-A tails. A number of many adenine, as you can see here, poly-A, many adenine nucleotides, a string of adenine nucleotides will be added to the 3' M. So this 5' capping and 3' and they are attached. These poly-A tail is also used by the recognition by the ribosomes. These are also used for the, when they will be released into cytoplasm, they are recognized by the ribosomes. They are also for the recognition by the ribosomes and also to prevent from the degradation by the exonucleases. The other process is splicing. Why the RNA splicing is required? As the RNA is synthesized, it will copy all the sequences, coding and non-coding regions. So the coding regions are known as exons and the non-coding regions are known as introns. So these intervening intron regions are removed by the process known as splicing. RNA splicing will remove these non-coding regions and the remaining coding regions will combine together by phosphodiester linkages. And now this coding region will be now having the code for the synthesis of the proteins. Now this code will be recognized by the ribosomes and will add up the amino acids for the synthesis of the desired and the required proteins. If you see here in this structure, this is now mature RNA which has the cap at the 5' end, that is the 7-methyl guanosine, and it has poly-A tails at the 3' end. At the 3' end, poly-A tail is attached. So the messenger RNA processing or the post-transcription modification includes capping, poly-A tailing, and splicing. In splicing, the intervening uncoding regions that were transcribed during transcription are removed, and then the coding regions will again combine together by the process by the phosphodiester linkages. So how this process is catalyzed? This is catalyzed by enzyme known as spliceosome. If you see here, this is the enzyme spliceosome which removes the introns and then combines together with the exons. The intron regions, the uncoded regions, are cut out by this enzyme known as the spliceosome. And this process is only specific for the eukaryotes. Once the mature RNA is synthesized inside the nucleus, they will be carried through these nuclear pores. These are the nuclear pores. They will be released by these nuclear pores. This messenger RNA will now move by these nuclear pores into the cytoplasm. Into the cytoplasm, they will be recognized by the ribosomes, and ribosomes, they will combine up the amino acids, and these amino acids will then lead to the formation of a specific protein. Once the pre-messenger RNA is fully processed in eukaryotic cells, it is now mature and can exit the nucleus through the nuclear pores to be translated into protein by the ribosomes. This was the whole details step by step by the transcription in eukaryotes. As the transcription in eukaryotes, it consists of the Preinitiation complex followed by initiation, elongation, and then termination. And after the termination, the RNA is premature RNA, and then it is processed. It is processed to form the mature RNA. This was all about the eukaryotic transcription. To understand the transcription process in prokaryotes, there is a separate video in which it is explained in details.